Hello, I'm Tracey Steig. I'm presenting a talk today called Use of 3D Acoustic Tag and Scanning Split Beam Hydroacoustic Tracking Techniques for Evaluation of a Wastewater Discharge Plume. First of all, I'd like to acknowledge my co-authors, Sam Johnston from HTI and Dave Thomas from Robertson-Bryan. This study was conducted for the Regional Water Quality Control Board, Central Valley Region, on behalf of Sacramento Regional County Sanitation District. This was a very large temperature study to assess the thermal impacts of this wastewater discharge. And it was very comprehensive. There were studies looking at the river temperature, effluent temperature, dissolved oxygen, river flow conditions, thermal plume mixing. In addition, there was hydroacoustic monitoring. There was acoustic tag monitoring of Chinook smolts and predators. And there was also standardized angling that was being conducted. Today's talk will only focus on the two techniques of scanning fixed aspect split beam hydroacoustic and 3D acoustic tag tracking techniques. This was conducted in the Sacramento River at the Sacramento Regional Water Treatment Plant diffuser pipe near Freeport, California. And this was a study conducted in 2012 between February and May. The primary study objective was to determine the thermal impacts on fish by this wastewater effluent from the treatment plant. But more specifically, the questions were asked, did the thermal plume from the treatment plant, number one, attract higher densities of predators, and number two, impede the downstream migration of the Chinook smolts, either through predation or from avoidance of the thermal plume. So this is just a top view looking down at the location of this diffuser, and this is in the Sacramento River. The river flows from top to bottom, and it comes from the east bank. The pipe does, and in the first 100 feet, there's no ports. The next 100 feet, there are ports, but they've been blocked off. And finally, where the ports are open are the next 300 feet. And this is just a picture looking across the river from where the pipe enters in the water, and it's the approximate location of where the pipe is, looking across the water. For the acoustic tag portion of the study, we used a model 290 acoustic tag receiver and 14 hydrophones. And we tagged 99 predators with the model 795 LG acoustic tag and 298 Chinook smolts with the model 795 LM acoustic tag. These tags are fully programmable for pulse width, period, and they operate at 307 kilohertz. This is a plan view, again, of the diffuser pipe, and the colored dots correspond to the 14 hydrophones that we utilize to track the predators and Chinook smolts in the area around the diffuser. We also did hydroacoustic sampling at the site, and we utilized an HTI model 244 multi-frequency echo sounder. It utilized two 6x10 degree elliptical transducers, and they were mounted on dual axis rotators. And we scanned these transducers horizontally in 14 different scans, as shown here on the screen, 7 on each of the transducers. And the minimum on-axis target strength was minus 40 dP, and these operated at 200 kilohertz, and we utilized a chirp signal. This technique is more fully described in Steig and Johnston 1996 publication. So I'll just jump into the hydroacoustic density results. This is from target tracking, knowing the volume of the sampling hydroacoustic beam, and then calculating out the density. So again, this is a plan view with the flow from top of the screen to the bottom, and at the top of the screen you can see the diffuser pipe. And now colors correspond to fish densities. with low fish densities being blue, medium fish densities being green, and high fish densities being yellow and red. And so for the first week of the study, you'll notice that there were relatively low densities of fish, but there were 3 areas of higher concentration, as shown in the bubbles here. In the second week of study, similar results were seen. Again, 2 areas of higher concentrations in approximately the same locations. And then finally, in the last week, again, you see these 3 areas of higher concentrations. But for the most part, these were relatively low densities of fish. And when this data was overlaid by the thermal plume and looked at where these fish were detected and what the densities inside and outside the plume were, there were significantly lower densities of fish found within this thermal plume itself. So looking underwater, this just shows, looking upstream, the diffuser pipe coming out into the river, and you can see the legs of the bridge upstream, and it just shows basically the 3 areas of higher concentrations of fish density. Now I'll go into acoustic tag position results of Chinook smolts. As I previously mentioned, there were 298 Chinook smolts that were tagged and released, and they were released in 4 different releases, approximately a week apart, and they were released 10 miles upstream. This is an animation showing the first 2 releases of 75 fish each going by the wastewater treatment plant, and the colors of the dots will correspond to the elevation in the water column, with red colors being near the bottom of the river, the yellows being near the middle, and blues being near the upper water column. And this is a very fast animation. In fact, it's approximately 11 hours of data per second as it scrolls by. So you can see the fish stream by very quickly, and then there's a pause, and there's a couple stragglers that come through, and then you'll see the second release group come through very quickly. And so that's 2 out of the 4 release groups that passed the diffuser. And so now I'll just show a summary of all of these release groups as they pass the diffuser pipe. And so for the most part, you can see that these fish show a straight path behavior, and they're primarily seen in the middle of the river with very few along the edges. Same thing for the second release group, again concentrated in the center of the river with very few on the edges and straight path behavior. And then finally the third release group, again similar type of results. So I just wanted to reiterate, this was straight path behavior, primarily in the center of the river, and we saw no predation events within the thermal plume during the study. So now I'll go into acoustic tag position results of the predators that were tagged. As I mentioned before, there were 99 predators that were tagged, and of those 99, 47 of those fish that were tagged, they were caught within the area of the diffuser, they were brought to the surface, they were tagged and released in the same place. 47 of those 99 immediately exited our acoustic array within 60 minutes and were never seen again. Of the other 52, those were seen multiple times in the array and over at least over 60 minutes in the array. But of those 52 fish that were seen greater than an hour, very few of them were seen in the array more than or up to 24 hours within the array. There were approximately 7 predators that were seen in the array for over 24 hours, and those consisted of 2 striped bass, 2 Sacramento pike minnow, 1 American shad, 1 smallmouth bass, and 1 white catfish. So out of all these 99, really there was only 7 that spent a significant amount of time in and around the diffuser pipe. And knowing the positions of all these predator fish and the amount of time that they spent in a given location, you can come up with what I call a position time density, because it tells you how long a fish was in an area over time. And looking at all 52 of these fish that stayed in the area, the densities are calculated, and the colors correspond to density, with lower densities being in the blues, medium in the greens, and reds being higher densities. And so you can see 5 areas where there was relatively high concentration of fish, but of those 5 areas, those only represent 7 fish that stayed in those areas. This technique is described further in Steig and Johnston publication in 2010. So if you look at it underwater, like with the hydroacoustic results, it shows 5 different areas of higher concentration. And as you notice, 3 of those areas are similar to the hydroacoustic areas of high concentration. So in summary, the split beam hydroacoustic showed areas of high density of predators, primarily in 3 distinct areas downstream of the wastewater discharge pipe. There were 99 predator fish that were captured and acoustically tagged and tracked in the area of the wastewater discharge pipe. Only 52% of those fish, acoustically tracked tagged predator fish, stayed in the area of the wastewater discharge pipe more than 60 minutes. And the position densities of these predators showed similar distinct areas of high densities as the hydroacoustic results. There were 298 Chinook smolts that were tagged and released and tracked in the area of the discharge pipe. These Chinook smolts primarily showed a straight path downstream behavior as they passed by the wastewater discharge pipe. And the conclusions, overall, the densities of predator fish were relatively low in the area of the wastewater discharge pipe. And this was seen in both the hydroacoustic and acoustically tagged predator results. The densities were consistently and significantly lower within the thermal plume as compared to outside of the thermal plume, this suggesting that the thermal environment within the plume was not concentrating the fish. There were no evidence of Chinook smolts avoiding or being impeded by the thermal plume. And finally, no tag predation events of the Chinook smolts were detected in the area of the wastewater discharge pipe. Thank you. Are there any questions?